President Ansai and other colleagues at JSPS, I'm very grateful to all of you for inviting me to join you for this particular event. And as you can see, the title of my speech is the Eureka Moments in Science. And science is, of course, as we all know, science and technology are the prime movers in our modern society and has been like that since the 18th century when rational experimental science started to develop. And since then we have witnessed an absolutely remarkable development in our field. Now, originally most of the science was based on formulation of hypotheses and then deductions from the experiments that one performed. But with time, there's been a development of a remarkable range of different techniques. And this has allowed the science to be formulated to concern using developed methods to straightforward questions. And one could refer to this as kind of vertical and horizontal science. And both of them are of great value, as I will exemplify. Sometimes there's a quantum jump in the advance of science, and sometimes also referred to as paradigmatic shifts. And in the will by Alfred Nobel, he used the term discovery. And the discovery is the word that we use when they guide us in the work on the Nobel Prizes. Now, I've been involved since a number of years back in writing books about Nobel Prizes. And 2 books have been published so far. I'm working on the 3rd book. It takes some patience because there's a 50-year secrecy period. But after 50 years, you can review the archives and you can see how the discussions developed and what the reviews were that underlied the choice of a certain new recipient of the prize. And the time that I've been mostly involved with then is the 1950s and the early 1960s, which is a very critical time in biology because that is when one started to formulate all the new concepts of what we call the central dogma that I'll come to in a moment. But the first example I would like to give is a prize to Frederick Sanger in 1958. And you would ask today, why was this an important prize to define the exact amino acid sequence in insulin? Well, at the time, it wasn't even clear that insulin was a homogeneous population of molecules, that they all had the same composition. And that was really firmly demonstrated by Sanger, that all molecules looked alike. But there was another shortage of knowledge, and I can see that in an investigation by the Swedish Nobel Prize recipient Hugo Tyrell, who reviewed Sanger in 1953. And the question he asked was, how could this be synthesized? Should there be individual enzymes that cared for the link of 2 independent amino acids, and how would that work? At this time, the whole concept of information molecule was not available, and only later did one understand that the peptide link was also of the same kind throughout the molecule, and there was something else that guided the position of the 2 related amino acids. So there were major quantum steps to be taken in order to understand this change. But perhaps the most famous discovery, of course, was the 1953 identification of the structure of DNA. And then all of you would think that immediately one would understand how things related to each other, but that certainly was not so. It took for the committee about 3 years to fully appreciate that, yes, DNA must be the information-carrying molecules. And the first nomination for a prize to Watson and Crick was not submitted until 1960, 7 years after the discovery. So this was such a large conceptual step to take that it took time to really assimilate it and to see the importance of it. So during this time and until the early 1960s, the central dogma developed with DNA as carrying the information and replicating with a high degree of fidelity, transcription into RNA, which was the molecule that could carry the information for the protein synthesizing machinery. And this was a very, very dynamic time. And conceptually, it was really revolutionary, because information molecules became really the name of the game. And it's characteristic that in the Nobel lectures in 1962, Francis Crick talked about the genetic code. Jim Watson talked about RNA, and only the third and the recipient talked about the structure of the DNA. But Sanger is unique in that he also received a second Nobel Prize in 1980. And now he had moved from sequencing protein to finding methods of sequencing DNA. And so you may ask, with such a two-timer, what was his secret? Why was he so successful in developing it? And if you read a recent biography about him, there is nothing conspicuous about, except that he had very free reins in pursuing his research at the famous laboratory of molecular biology at Cambridge. And there were years when almost no publications were emerged from his work. But he was very focused and, of course, technically very, very skilled, and he stayed with his problem and had this remarkable double success. But having now a technique to sequencing and to read the books of life, to sequencing DNA, our science in biology was completely transformed. And one could give a 1 hour lecture about each one of these points that are here on this little list here. But let me just mention briefly. So the first sequencing of a whole genome of an organism was in 1995, of Haemophilus influenza by Craig Venter and Ham Smith. And very quickly the techniques developed, in particular the so-called shotgun technique, which originally was believed could not be applied to very complex materials, but turned out to be really the key procedure. And with time now, there's been an enormous development of the technologies to read DNA and to take out DNA sequences. What are the consequences? Many different consequences. Of course, being anthropocentric, we think the human genome is very central in this. 2001, there was the first description of human genomes. We can look at the predisposition to disease development, drug metabolism, forensic matters. We can look into our history. The scientist Svante Pääbo is now looking at bones that are 40,000 years old. And with remarkable technical advances, one can now interpret what the genome looked like. And also, as many of you know, the Neanderthal genome has been identified and characterized. And it's been able to prove that, yes, with the modern humans did interact with the Neanderthal Neanderthal primates. Now, what else in determining of genetic relatedness? We can now look at evolution and really see the mapping of this, which is absolutely dramatic, because we can see how things relate. And of course, in Sweden, we are very proud of Linnaeus' categorization of plants based on the binomial taxonomy and on the morphological characteristics. When one used the modern techniques now, only 1 of 24 classes that Linnaeus had described has survived into the present time. And thereby, of course, also the first Japanese flora that was made by Linnaeus' pupil Thunberg, studying plants in the Shema in the Nagasaki Bay, is equally irrelevant today because we have all this new evidence. We can identify unknown forms of life, and what is emerging is that we completely underrated the diversity and the richness of life in what we could call invisible life. That is the microorganisms and viruses. And I could speak at length also about that, because it's completely transforming our concept of life. take the open sea. So 1 milliliter of seawater, you have 1 million microorganisms. You have 10 million virus particles. We can now characterize what they are. Most of them are completely unknown to us. If we start to multiply the amount of water on Earth with the number of virus particles, we end up with a figure of 10 up to 24. That's 10 times more than the stars of the universe. What do these viruses do? They are very critical ingredients in the whole ecological balance that we have. And it's a lot to be learned about this and in various contexts. Also in our own body, we have 10 times more microorganisms than we have our own cells that build up our body. What do they do? The changing perspective is that whereas previously when we got to learn to know about microorganisms and viruses, they were things that caused evil. What we're learning today that on the contrary, they are actually contributing to the way that we can manage our lives. And one can simply state that without the viruses, we would never have come to existence, human beings. So this is an extremely rapidly developing field. There are new techniques also in this. I just mentioned very briefly this CRISPR technique, by which, and using now a defense system that microorganisms have against certain viruses, we can now zoom in and exactly change in the genome a defect. One has managed research at Harvard to amend the germ cells in mice in which there are inherited genetic diseases and amend them. Discussion is now, what about applying this to humans? What about what the ethics of this? The irreversible change of a genetic defect certainly will be applied, but there's room for discussion. But the technique is absolutely amazing. Which are the three largest conundrums if we look at science? It's of course the origin of the universe. Now that's something for a physicist to comment on, but still we have the dark matter, 23%, dark energy, 68%, which we don't know the nature of, and therefore we need to know much more. That's much beyond the Higgs particle that was recognized by the Nobel Prize last year. Origin of life, of course, intriguing to reflect on that. We discuss a lot the so-called RNA world. Again, viruses play a major role. There are RNA viruses which probably reflect the time when the DNA storage form of information was not available. And on we go. And the third, of course, the origin of consciousness. Nothing is as tantalizing as trying to understand how we think and how we store information. The information is really a key word in this, and I very briefly would like to comment on how nature has solved this. So in immunology, we need to have an immune defense that can identify any kind of structure that is out there, any virus or any bacteria, whatever. And it was Paul Ehrlich speculated about a locking key mechanism, but it was intellectually very difficult to understand how could a key really develop its own lock. And the very visionary virologist, immunologist, Macfarlane Burnet, instead offered another proposal. That is that the immune system should work by selection, a clonal selection. And that was a theory of his, later proven by Tonegava to be correct, that during embryonic differentiation, there is a reshuffling in the genes. And that allows us to have in our bodies a resource to identify any chemical structure. Not perhaps with a absolute high precision, but enough to identify it and then to, by a microevolution, have a development. So in immunology, it's a selection of a pre-existing, a huge number of populations of different cells that can be used as antibodies. What about the olfactory sense, the most primitive of our senses, where we sort out molecules? Well, there's no way we could have individual cells and receptors for all the different molecules that we meet. There must be some other system. And we do invest quite a large percentage of our genome, more than a percent in human, and in mice perhaps 2-3% of the genome in different receptors. But the system is amplified by these different receptors, several thousands of them. They can react differently to different triggering molecules, and then the brain can integrate the pattern of reaction. So it's a combination of a large number of receptors and a combinatorial approach in the brain. But what about our own brain? Well, our own brain must be that information is taken from it out and integrated. And of course, understanding memory and the short-term and long-term memory is a fantastic challenge for the future. We know that long-term memory requires protein synthesis, but for the rest, we're very ignorant about this. But since, of course, there's no way by which we could have a brain that could include all the potential thoughts and references that there could be from the dawn of mankind to the exit of mankind, we need to have an instruction set. How does it work? That is an interesting, challenging question. New techniques now developing, studying the brain. Optogenetics is a fantastic technique by which you tag specific molecules with things that can fluorescence as you shine with light on it, and we can make brains in experimental animals translucent, and we can now study the tracks, giving them different colors, and really start to understand how it all interrelates. The DNA sequencing technology and the identification of all the potential proteins that can be produced by the human genome has led a Swedish group under Mattias Erling to try to produce antisera. This is now proteomics on a very huge scale to produce antisera to all the different human proteins. And that study is essentially complete and just published, a good part of that in Science. This is, of course, a treasure. There must be a fantastic number of reactions that allows further research in this whole area. But, so we talk about reading the books of life. What about writing the books of life? Well, Craig Venter and his group has continued this work, and they have been successful in synthesizing with absolute fidelity a DNA that contains more than 1 million nucleotides. This is it's copied the genome of a special mycoplasm, and one has managed to have that synthetic genome expressed in a mycoplasm in which one had incapacitated the existing DNA. So what is driving this whole cell is the synthetic DNA. This is indeed to create life in the form that we can do it today. But one can take it one step further. Then knowing the exact number of genes, and one can start to identify precisely all the different functions. We would like to understand the complexity of this system on the level of individual genes, and that is precisely what's being done right now. This is one of the most challenging dimensions for the future, namely to understand how is it all orchestrated. How does this orchestra without the conductor function? And there is still a lot to be learned in that area. But the techniques are there now to study in a synthetic cells the role of individual genes. It will not be a simple picture for sure, and there'll be a certain redundancy where gene products can compensate for each other, but we're learning amazing things. I wanted to take one final example also from the field of medicine. In 1985, we gave a Nobel Prize to Joe Goldstein and to Michael Brown. A fantastic prize, because they started out by learning from nature. They took rare families that had a defect in their capacity to metabolize and turn over cholesterol. And they identified the whole mechanism, a very, very complicated mechanism, a series of mechanisms in the liver by which receptors play a very critical role. And this then led to an understanding of the various processes. So the way we balance our cholesterol metabolism extremely critical, because cholesterol is a key ingredient in our cellular membranes, and the body must read out how that is balanced against the the amount of cholesterol that's added to the organism that is synthesized by the mechanism. So, and a lot of factors, there are genetic factors. You can see a difference between Asia and Europe. You can see how this affects heart infarcts. And today we also have drugs that can tune the level of cholesterol. The drugs that actually were discovered by a Chinese colleague by the name of Akira Endo. And all this is changing our life. There is no reason in the future that someone should die from a coronary infarct with all this knowledge. So, to finish off, what are the conditions that could further Eureka moments in science? Well, choose an important problem would seem to be critical. Crick always emphasized only if you choose big problems can you get big answers, which of course is true. But he also said, try to work with the simplest possible system in order to answer the question. Be heterodogmatic and bold. Learn from nature. I mentioned Brown and Goldstein. There are nature's mistakes of genetic defects that you can learn from, and unravel by the tinkering of evolution. Of course, you need to be well informed about advancing technology and means of information storage, processing, and retrieval. But then, so when I write this book on Nobel Prizes, it stirs a lot of reflections on individuals and environments and the conditions under which science can advance. It's striking that very often people who receive Nobel Prize have had mentors that have been previous Nobel laureates. There's a lineage of knowledge here that comes along. So there is a very critical social dimension in science, is to combine with the idea that you have as a scientist. And Peter Medaway referred to this as obsessionalism. You have to be obsessed. You have to believe in what you're pursuing and think that you're right. Many times it'll turn out that you'll be wrong, and that is the other flip side of the pursuit of science. And then you need to have, in fact, some luck and be aware of the role of serendipity. I think it was Pasteur who said that the luck favors the prepared mind. And so you have to be very, very open to what the fundamental in this is that science is not a predictable venture, and still we're making this progress using existing knowledge to advance new knowledge. And my final slide is actually a quote from Arne Tiselius, a Swedish Nobel laureate of 1948. But the year before, he was involved in Nobel work, and he said the following: when a new thought is born, or when one of the deep secrets of nature yields to a searching scientist, in this very act of creation, there is a pure and primitive happiness, deeper than anything of this kind which can ever be granted a human being to experience. And I think every one of you who are scientists have been through this moment when you are the only one, on the first one in the world, to see a piece of knowledge. That is the moment of happiness. And I need not emphasize that, of course, people do not pursue this thinking and their work because they want to get prizes or anything like that. They do it because they are curious. So a prize is merely a confirmation of work well done, but it's not a driving force. And that is what Tiselius is also saying in the final part here. In any case, we do not believe that to you, and these were the Nobel laureates, even the highest award, the most wholehearted recognition, can be more than a faint reflection of the deep satisfaction you must have experienced of your work. And this actually, as we heard the first speaker today citing Negizzi, that more the same line of thinking. So with this, I thank you for your interest. And